Okay, so first I want to thank the organizers, especially Kristen O'Brien, for inviting me. I also want to thank Julie, who did an amazing job with all the organization and sent me a box with a shirt and a Snickers bar, which I've been enjoying over the last talk about COVID. So thank you, Julie, you're amazing. And then I'm also looking forward to talking with the students for a couple sessions after this. And so we're going to transition to a very different topic from COVID. We're going to go from the pandemic to a bit more Zen place in the rainforest. And so I'm going to talk about my work on poison frogs and the evolution of parental behavior. And but before I get started, I want to, I wish I could have visited Alaska. I've never been there, and it's always been a dream of mine. But I'm sitting here in Stanford, California. And so I also want to acknowledge that our research at Stanford University occurs on the unceded and ancestral lands of the Muwekma Ohlone people. Okay, so if we think about social behaviors that all animals display, we can come up with a few different categories that are conserved across all animals. And so the examples, I hope you can see my mouse, that I have here are competition over either resources or mates, where aggression is typically displayed, or reproductive behaviors, where you have to secure a mate and perform reproductive behaviors to create offspring. And so these are all behaviors that are mostly conserved across animals, and I spent my graduate work studying the neural basis of these behaviors and how not only are the behaviors are conserved, but the brain mechanisms that promote those behaviors are conserved too. And as I was finishing up my graduate work, what I wanted to instead transition to is what I'm calling evolutionary innovations in physiology and behavior. So instead of studying something that's conserved across all animals, I wanted to study behaviors or aspects of physiology that were novel or new. And so some examples are predator-prey interactions. So this is a newt that is local to our area that is chemically defended, as well as the bombardier beetle. So they've come up with new mechanisms to defend themselves from predators. And then also aspects of social behavior that are evolved in some species, such as parental behavior. And it's this aspect of parental behavior that I want to talk about today. So my lab in particular is interested in the evolution of parent-offspring interactions. And so we specifically focus on parental care because it has evolved many times independently across animals, and it can lead to really interesting conflict as well. So parent-offspring conflict. And what I mean by that in an evolutionary sense is that parents have to decide how much resources they want to allocate to their current versus future offspring. And so, as I said, these are shaped by resource variability. So there can be not only interactions between the partners and between the children, but also between the children and also between the parents and the children. And so there's a lot of interesting dynamics that happens in a family unit that my lab is very interested in understanding. And what I'm going to talk about right now is the neural basis of parental behavior. And I became very interested in this question while I was rearing my own offspring. And so we know quite a lot about how the maternal brain reorganizes to promote motherhood. And so we know this because of really elegant decades of work in laboratory rodents where moms care for their pups. And so, but around the time I was having my own kids, I started to wonder a lot about what was happening in the brain of my partner. And so, just kidding, but you know, what is changing in the brain that allows some males to care for offspring in some species, but not others? And so in mammals, what's happening, and especially in biparental mammals, is that you have the male pair bonding to the mate around the same time as the offspring are being reared. And so to me, that was a very hard question to disentangle, meaning that both pair bonding and parental care was happening in the brain at the same time. And I was specifically interested in fatherhood. And so what I decided was I needed a model system that enabled me to address different aspects of parental care, regardless of sex and pair bonding. And so I dug deep into the literature and looked at how the evolution of parental care across animals, which again has evolved many times independently. We see it in vertebrates over here, all the way to mammals. And what I have here is the female uniparental care on the bottom, biparental care in the middle, and male uniparental care on the top. And so you'll note that male uniparental care is not present in mammals due to obligatory lactation. And then it also has never been described in reptiles. So what that left me with was studying parental behavior in a clade of amphibians that were all very closely related and have very similar ecologies. So they were in fact the only clade that I could find that fit all of those criteria. And so I decided that I was going to spend the next decade of my life developing poison frogs as a model system in neuroscience and physiology. So everyone knows what a poison frog is, but I want to point out a couple of traits that you might not be familiar with. And so they're famously brightly colored. You've probably seen them in museums or in a Nat Geo show. But what you might not realize is the extent of the variation in color. And so these three frogs here, for example, are actually all the same species. They're just in different locations or populations in Colombia. And this variation is due to a mix of mate choice and predator selection on phenotype. And so there's a lot of variation in color and patterning. And this coloration advertises that they are toxic, that they are not a tasty snack to predators. But what you might not also realize is that there are quite a number of poison frog species, meaning like in the same poison frog clade, they're actually quite drab and brown and not toxic. And so they're not like famous like their colorful cousins, but we love them because they provide a really nice comparison to their colorful, toxic, closely related species and allow us to do a lot of cool physiological and evolutionary studies. And third, and what I'll focus on today, is that they are amazing parents. And so in each of these pictures I'm showing you, the frog has a tadpole on its back, so it's carrying its tadpole to water. And in some species, like this one right here, they have extended egg provisioning behavior. And so for the next little bit, we'll take a deep dive into these behaviors and the neural circuitries that promote them. So we'll answer, what is the neural basis of parental behavior and how does ecology drive these behavioral adaptations? So first, I would like to start off within a more phylogenetic framework because I think it gives us a lot of power towards addressing evolutionary questions in this system. And so first, so this is the family Dendrobatidae, or South American poison frogs. And so what I want to point out a couple things on this phylogeny. One is that toxicity has evolved at least 4 independent times. So these gray boxes are all toxic species. So you can see that there are many independent transitions to this phenotype, that most of the toxic frogs have a dietary specialization on ants. This is actually a new species of ant that we named and recovered from one of the stomachs of our frogs. And then if we map behavior onto this phylogeny, what I want you to notice is that male uniparental care is ancestral on the phylogeny. And then we have 2 independent transitions to different modes of reproductive behavior, including female uniparental care and biparental care. So these toxic frogs in this toxic clade here have evolved other parental care systems. And so I think many people are surprised that frogs have parental care. And so I want to describe to you a little bit about what that looks like. So this adorable picture is a male frog guarding his clutch. So they have to keep them moist, and they, if they leave them, they could be eaten by egg predators. So not only do they keep them moist, but they also guard them. When we remove males from their clutches, they get eaten. And so he has a very important job here. But so the egg clutches are reared on leaves on the forest floor, but tadpoles are aquatic and they need to get to water somehow. And so the parents do this piggyback ride behavior, this tadpole transport behavior, where they take the tadpoles to pools of water. And in some species, they do this extended egg provisioning behavior as well, which we'll also get into in a bit. But what's special about this clade, again, is that the sex performing these different parental care tasks differs among closely related species, so we can compare behaviors and neural mechanisms to try to understand what are core aspects that promote parental behavior in the brain. Okay, so the species I'll be focusing on today, and the ones that we do a lot of field and lab work with, are these species here. We have a monogamous and biparental frog species, a female uniparental care species, and a male uniparental care species that are all pretty closely related. And so I'm going to tell you a couple of short stories. One about the neural basis of piggyback rides is the first thing we'll talk about. Then we'll talk about this egg provisioning behavior. And then if I have time, I'm going to link this back to ecological resources in the environment and how toxicity and parental behavior are very tightly linked. Since there are also a lot of students in the audience, I wanted to start off with a general framework on how people who study behavior in natural context really think about scientific questions. And so these are kind of the famous 4 questions that you think about when studying animal behavior. And these were put together by Nicholas Tinbergen, who won the Nobel Prize for formalizing the study of behavior. And so he broke these down into proximate mechanisms, or how things work. And this could be questions like, what are the neural mechanisms of behavior? Or how are behaviors acquired or learned during development? So mechanism and ontogeny. It could also be divided into more ultimate or why questions, like what is the adaptive value of behavior and how does this behavior evolve and diversify? And so we're going to come back to this framework as I tell you these 3 different stories as we kind of fill in why we think parental behavior has evolved and how that has happened. Okay, so let's start off with the neural basis of piggyback rides, or offspring transport. And so we're going to get into the mind of a frog for very briefly and talk about the cognitive demands of tadpole transport. And you might think that this is a very simple behavior, but the frogs actually have to make a lot of interesting decisions when they're doing this task. And so as I mentioned, they lay their eggs on the leaf litter where the tadpoles develop. And when the tadpoles hatch, they have to get a piggyback ride to a pool of water. Now, the rainforest is a complex place, and they have to remember where high quality pools are located, because if you put it in a low quality pool, the water might evaporate and then the tadpoles will die. And the tadpoles have a pretty long development period, which is for about somewhere between 2 to 6 months. And so it needs to be a pretty stable pool that they're choosing. This is an important choice. And they have to do it quickly because as they usually deposit their tadpoles outside of their territory, and while they're gone, they might have, they risk infanticide by competitors. And so this has been really nicely described by Ava Ringler, where when males take over territories, the first thing they do is they eat all the eggs of their competitors. And so this is an important task for offspring survival, and they need to do it quickly, but they need to make good choices while they're doing it. So what we wanted to ask then is there a general parental care circuit in the brain that's the same across males and females, or are there sex-specific mechanisms? And so what we wanted to do is we wanted to compare these 3 different species with different the parent who was doing this tadpole transport behavior differed. The sex differed across species. And so what we wanted to do is we first wanted to ask what brain regions are involved. This might not surprise you, but not much is known about the amphibian brain. And then we wanted to know what neuronal cell types are involved as well. And so when trying to decide where to look in the brain, what we decided to do was look for brain regions that are also present in mammals and have been linked to some type of social behavior. So there are different networks in the mammalian brain, like the reward system, which analyzes valence of stimuli, and so the social behavior network. And so these brain regions are present across mammals, and they have very distinct features where they have different cell types and connectivity in these different brain regions. But what's important is that these brain regions are very old, and they're present across all vertebrates. And so one of the things I did when I was a graduate student is I looked at these brain regions across mammals, and I made a map of where these are all located in every other vertebrate taxa so that we could have meaningful studies about the neural basis of behavior in a fish or a frog and be able to translate or inform how these things work in mammals as well. Okay, so the experiment that I'm going to tell you about that looked at tadpole, the tadpoles don't actually float here. I was afraid you wouldn't see them. But anyways, we're going to look at a tadpole transport experiment where we did this in the lab. So we house males and females in pairs, and they have a bunch of foliage. We have a frog colony with like 500 frogs down in the basement. And then they have a little pool with which to transport that they could transport their tadpoles to. And so we did these both in the lab. We waited for them to transport their tadpole, and then we sacrificed both the animal that was doing the tadpole transport and their partner who was not doing transport. So we did these in the lab. We also did a set of experiments in the field. And so we do a lot of field work in Ecuador, where we work with local Ecuadorian scientists and conservation biologists to maintain an array of enclosures where we can do some behavioral experiments. And so the experiment that we did is we compared animals that were not caring for offspring to animals that were doing tadpole transport behavior. And the assay that we did is we did an immunohistochemical reaction, so using antibodies, to detect phosphorylated ribosomes. And the reason this is a reflection of neuronal activity is that when neurons become more active, they need to make more protein and they phosphorylate their ribosomes. And this is a change that we can detect with antibodies. And so what that actually looks like in a frog brain is that when we do the stain, it labels active neurons brown. And so then you have to count the cells, which was done by my fabulous postdoc, Ava Fisher, who now has a job at the University of Illinois as an assistant professor. And she counted in all these different brain regions that I previously mentioned are linked to behavior. And so I'm going to show you one example here in one species. This is my favorite species, this blue guy. And the symbol over here will tell you which sex does the tadpole transport behavior in that species. And so what I'm going to show you on this brain over here is I'm going to color brain regions that have more active neurons when the frog is doing tadpole transport behavior. And so what you can see is that there are a lot of different brain regions that have more neurons on when the parent is doing this tadpole transport. And so, we could stop there and say, oh, look, these brain regions are contributing to tadpole transport behavior. But again, the reason why we started this model system in the first place is that we wanted to do comparative work. And so these are the results in the two other species, and you can see that there are quite a lot of differences. So, for example, this brain region here is active only when males are doing the parental care task. There are some brain regions that are only active in monogamous species. Interestingly, this brain region is also linked to monogamy and biparental care in mammals. But today we're going to focus on the similarities and core brain regions that promote parental behavior. And so these are the medial pallium, which is the frog version of the hippocampus, and the preoptic area. And so I'm just going to show you the actual data here real quick. And so what I'm showing you here is these are males and females that are not parents, and then these are animals from the tank that was doing tadpole transport behavior. But remember, only the male in the species does tadpole transport behavior. So I'm going to put a box around him so that you know who's doing the transport task. And so you can see there are more active neurons in the preoptic area of the male doing this tadpole transport behavior. And this is also very similar across species. We only have females in this species because we collected them in the wild and we are not quite sure who dad is. And so the other things I want to point out is that these two, the mom and the dad, even though the dad is doing the tadpole transport here, the mom still has neural activation, pretty high neural activation. These two are actually not statistically different. And we think that this is because they actually coordinate care in this species, and both males and females participate in offspring care, unlike the other species. And so this is the preoptic area, which is part of the hypothalamus. And this is the hippocampus, which shows similar things. So I'm just going to briefly point this out. So the hippocampus in mammals is linked to spatial cognition or space use, among many other things. And so what I want to take a brief moment to do is link this behavior back to ecological resources. Because I talked about how these frogs need to find water pools in their environment, and spatial memory has been found to be very important in this task. And so what we're doing now, I have a postdoc in my lab, Andreas Pasikonis, and so he studies spatial cognition in these frogs and how they move throughout the rainforest. And so what we've developed is these these frog pants. I guess they're more like frog songs maybe, but in any case, they have these little receivers attached to them where we could follow them around in the forest and see how they make behavioral decisions. And so this is ongoing work. Okay, so at this point, we've localized the brain regions that are important in a behavior, but we don't know what cell types are important in that behavior. And so one of the reasons that we do use phosphorylated ribosomes as a proxy for neural activity is because we can immunoprecipitate the ribosomes and pull the mRNA associated with the ribosomes down in an immunoprecipitation reaction and sequence the RNA with an Illumina machine. And so what that does is that instead of sequencing all the RNA in a brain region, what we're doing is we're sequencing the RNA from active neurons. And so they're more likely to be associated with our behavior of interest. And so we did this in both brain regions, the hippocampus and the preoptic area, in males that were transporting a tadpole versus not. And so I want to explain what this phosphotrap data, so this technique is called phosphotrap, where you're pulling down phosphorylated ribosomes and sequencing the RNA associated with them. And so I want to explain what this plot will look like. So we do a ratio of the immunoprecipitated RNA and the input RNA. And so all the dots at the top here will be transcripts enriched in transporting males, and this is a marker of active neurons. Because we're comparing animals doing the tadpole transport to control animals, we can see that transcripts that are down here in the negative realm are depleted in transporting males, and this is a marker of silenced neurons, or neurons that are not as active during tadpole piggyback ride time. So there's a lot of RNA generally happening in these cells, but what I'm going to point out are particular cell type markers, like a dopamine neuron or an oxytocin neuron and things like that. And so these are the orange dots here. And so there are a lot of different markers that came up. So here's the androgen receptor, for example. And so how we interpret this is that cells that are active during tadpole transport tend to have the androgen receptor, tend to be androgen receptor positive. And so that tells us something special about the neurons that we think are promoting these behaviors. So this is the hippocampus, and we also did this in the hypothalamus. And I'm showing you these two side by side. They're different brain regions, and you can also see that the results differ between the two brain regions. And that's true where we do have quite a number of different cell type markers. But when we look at the overlap, there's actually just one overlap that was significant in both brain regions, and that was this neuropeptide called galanin. And so when I first found this, I was a little disappointed or not interested maybe, because galanin has typically been associated with feeding behavior. And so it's associated with, in the context of feeding and diabetes and things like that. But it also has been associated with paternal care. And so if you think about male laboratory mice, if you present a male mouse with pups, they will eat them or commit infanticide. But work in Catherine Dulac's lab, and Catherine Dulac actually just won the Breakthrough Prize for this work. If you stimulate galanin neurons in a male, it like switches them from infanticidal males into caring dads, like with this flick of a light, which is pretty amazing. And so what we then wanted to look at, we were like, okay, so we're thinking about our frogs, they're doing this egg care. We know that when they take over new territory or when there's low food resources, they do this infanticide behavior. So, you know, maybe what's happening in our frogs is that that galanin neurons are on when they're doing this tadpole transport task. And so the other thing that we were excited about is that galanin circuits are quite similar in male and female rodents. And when you activate them in mice, you can promote parental behavior in both sexes. And so what we thought was maybe there was this link to this conserved parental care circuitry, meaning that the circuitry that promotes parental behavior is the same in males and females, and then what activates the circuit is what's different. And so to test this idea, we did a co-stain to look at the number of galanin neurons that were active during different tasks in our frogs. And what we found is that during tadpole transport in the biparental species, that there was this increase in galanin activity during the parental care phase compared to frogs that were not caring for offspring. So we found this in one species. Interestingly, galanin neurons weren't active in the other species that we were able to find, which I think points to some interesting species differences, mainly that both parents in the biparental care species are taking care of offspring, similar to other species where galanin has been linked to parental behavior. And so maybe it coordinates both parental care in coordination with partners. But what I want to point out, and this is links back to some broader theme, is that there seems to be this modification of feeding circuits when you evolve new social behaviors. And so this has been shown in many different species as they transition from singletons to either paired or parents, and as species evolve towards solitary to more social. Okay, so just to summarize the part that I told you so far, that we see overlap in neural mechanisms mediating tadpole transport across species, but there's still evidence for species specificity at the neural level. So there were a lot of other brain regions that were active that weren't linked to these core brain regions. But that overall, it looks like this modification of feeding or energy circuits is involved in the transition to parental behavior. And this is an idea that we're continuing to follow up on. And so if we come back to this 4 questions idea, what I told you so far is that galanin, at least in some species of mammals and frogs, seems to promote parental behavior. And that because we were able to look at a bunch of different species that where different sexes participated in parental care, we were able to identify some shared neural circuitry underlying tadpole transport. And so the comparative aspect was really important in this study. Okay, I'm going to briefly talk about my favorite behavior, which is egg provisioning behavior. And it is very cool. And so it'll be a little bit briefer, but it's super cool. So this tadpole provisioning is a costly behavior. And so in species where only dads are involved, they do this egg care, they do the tadpole transport, they put them in a water pool, and then care here stops usually. And then the dads go back to their clutches, and this cycle is repeated and repeated and repeated. When moms are involved though, they really go an extra step here. And so let me explain to you what female uniparental care looks like in these species. Moms do the same thing. They take care of their clutches, they do this tadpole transport, they transport each tadpole to a different nursery. And so they're often taking care of 5 to 9 tadpoles at once. They all get their own nursery. And then what happens is that the mom has to remember not only to take them there, but also where she put them. And so she puts them in these plants that have little pools of water in them in the rainforest. And so she has to remember where they put them because she comes back to visit them about every 3 days to feed them. And so this behavior lasts about 2 to 4 months. And so it's this really extended provisioning behavior where she puts a lot of energy into rearing these tadpoles. And so we're going to take a little deep dive into this very cool behavior, this egg provisioning behavior. So when I first saw this, I was like, what? Why? Like how and why do they do this? This doesn't make sense. This is so costly. And so what we decided to do was was to take a look outside of South America for a moment. And so the South American poison frogs I told you about, they have defensive chemicals, they have these flashy colors, they have parental behavior. But what I haven't mentioned yet is that not only have these traits, this suite of traits evolved in South America, but they also evolved in Madagascar, the island of Madagascar off the coast of Africa. And so these are Mantella poison frogs. And so these are about as far apart as you can get in the frog world. Here are different species. They're bright and colorful, they're toxic, and they do this egg provisioning behavior. And so except one species, Dendrobatids are here and Mantellas are here. So in hylids and rhinids, the 2 major branches of frogs. And they're about 150 million years apart. So it's about as far apart as you can get in a frog. And so what we decided to do was to go to South America and to go to Africa to study egg provisioning behavior in this instance of convergent evolution. And so this was done by 2 postdocs in the lab, Alex and Ava. And so they looked at behavior. We want to take the same approach, do behavior, look at brain regions, and look at neurons underlying this behavior. And so just to give you a little bit of background about what this behavior looks like. And so moms provide these unfertilized trophic eggs to tadpoles. And so it's kind of like the frog version of lactation. It's this physiological production of food for offspring. This benefits the offspring. They're more likely to make it past metamorphosis. And the tadpoles beg their mothers for meals, much like a baby chick does. And so I'm going to show you a video of this right here. This tadpole is nipping at its mom and doing this little like dancing behavior. I hope this is coming through on Zoom. I'm sorry if it's not. These videos are also all over YouTube. And so what the tadpole is doing, it's communicating to mom that it's hungry. The tadpole does not dance if it's satiated or if it's not hungry. And so it's considered an honest indicator of need, much like in chicks or crying baby humans. And so there's been some studies that suggest that these tadpoles also carry toxins and that it allows for the exploitation of novel habitats. And so I want to bring this behavior back to ecology for a moment and talk about resource limitation in parental care. So if you're a poison frog and in the rainforest, you have a lot of options for pools. You can either put your tadpole in a very large pool where there's lots of resources, but also lots of predators. Or you can put them in a very tiny pool where there are no predators, but there's also nothing to eat. And so how frogs have responded to this is that they've evolved these different reproductive strategies that fit the needs of the tadpoles in the pools. So these frogs do, I don't know if you can see this, there are a bunch of tadpoles on this frog. This species does the school bus method of tadpole transport. It drops all of its babies into one pool, one huge pool, and they all get along. These tadpoles over here, moms drop them off into like 10 mils of water and then need to come back and feed them because there's nothing else to eat. And so the tadpoles, you think gross eggs, but these are little protein packets, and the tadpoles grow much better on these eggs than they do in like synthetic food that we feed them. So the eggs are magical in some way that we don't understand yet. So we did the same assay for behavior in this experiment that we did in the last experiment, except instead of looking at closely related species where they differed a little bit in which sex was doing the behavior, we're looking at the same behavior but across a wide evolutionary distance in this egg provisioning behavior. And so the results are here. There are some differences again, but I want to just point, for the sake of time, point out the similarities, which is this lateral septum area, and again, this hypothalamic area here. And I'm going to zoom in on this hypothalamic area. So there's increased neural activity when the moms are doing this egg feeding, and you can see the micrographs here. So these are slices of brain, and the brown neurons are neurons that are on during nursing. And so this is a non-nursing mom, so you can see the difference. Now, what was exciting to us about this is that in this brain region regulates maternal care in mammals as well. And so what we thought was like, oh, and that oxytocin neurons are also present in this brain region. So we were like, oh, maybe oxytocin plays an analogous role in frogs as in mammals. And so we set out to test this really beautiful hypothesis. And co-stained oxytocin and phospho-S6, so phospho-S6 being our marker of active neurons. I was pretty sad when I looked at these results, and let me show you why. So these are the percent of active oxytocin neurons. And so there are a lot of oxytocin neurons that are active in our African frogs here, but nothing is happening here in our South American frogs. And so I think this is a good example of how you have to let go of your beautiful hypothesis and listen to what the data tells you. Because what we basically found, and I'm going to summarize this on the next slide, is that there are these shared neural mechanisms that probably promote egg provisioning behavior. So we found these, even though these frogs are 150 million years apart, we found these same brain regions that are on during egg provisioning, but that we found that different neurons actually promote the behavior. And so it's probably oxytocin neurons in one species, but not the other. And so just because you have an analogous behavior does not mean that the neural mechanisms underlying those behaviors are the same across species. So meaning that they have an alternative solution for promoting maternal behavior in the brain is our current idea, which is a pretty cool idea, I think. Okay, and so I'm going to spend my last 5 minutes or so coming back to the question of why this costly behavior has evolved. I mean, this behavior is incredibly costly. Why are they taking care of 5 to 9 tadpoles over many, many months? And so when we look back at this phylogeny that I introduced you to in the beginning, what you see is that egg provisioning, or when moms are involved, evolved twice, but they evolved in toxic frogs. And so we thought maybe something about being toxic allows for the evolution of egg provisioning. And so we wanted to test this idea a little bit, which gets into a little bit about chemical ecology. And so briefly, I'm going to talk about we do a lot of work on toxins in my lab, maybe next time, or we can talk about it later. But they carry these small molecule neurotoxins on their skin. Some of them are lethal, some of them are not. I don't work on any lethal species, I promise my children. But these small molecules are called alkaloids because they have these ring structures on them that have a nitrogen group on in the ring. That's what makes it an alkaloid. And so these are small molecules, not peptide toxins like you would see in a rattlesnake or a cone snail or something like that. And so the frogs carry these compounds on their skin, but what I haven't told you yet is they actually sequester these from their diet. Poison frogs do not make their toxins on their own. They have to get them from a dietary source. And so all of the frogs in our colony, for example, are non-toxic unless we make them toxic for some reason, which a number of grad students do frequently. But what this links back to is the ontogeny of toxicity. And so in adults, when you're running around in the leaf litter, you have lots of access to the animals that they actually get their toxins from, which are ants and mites in the rainforest. Tadpoles, on the other hand, are in the water. They do not have access to the animals that have alkaloids. They eat algae, they eat larvae, mosquito larvae, and they sometimes eat each other. And so, but what this all means is that poison frog tadpoles are supposed to be non-toxic. But I mentioned before that there is some evidence that in egg feeding species, the tadpoles also have toxins. And so what we wanted to do is we wanted to see if this was a possible, another adaptive advantage in addition to nutritional resources. And so what we wanted to do is we wanted to see, well, they might get these toxins from the eggs, from their mothers, or from the water. And the experiment that I'm going to show you does not disentangle these two things yet. We are working on that. And so the samples that we collected were moms, tadpoles, internal eggs, so eggs that the mom hadn't laid yet, and trophic eggs. And this was done by one of my students, Nora Moskowitz. And so what I'm going to show you is this is all the toxins, 100%, that is present in moms. If we look at the oocytes, we see that there are also these toxins present in the oocytes, which means that the eggs are loaded with toxins prior to deposition. The tadpoles are also toxic, and so are the eggs that they feed them. And so, as well as the water, which we think might help defend the water resource from other animals. And so this was pretty cool. This was the first example of where we linked the provisioning of toxins to tadpoles through the eggs that they were being fed. And so here's just a box plot of these toxins so you can see kind of the distribution. Okay, so, but if toxin provisioning is an adaptive advantage to this egg provisioning behavior, then it should also be found in our Malagasy frogs. And so we tested this idea using this independent replicate that nature had provided us. And what we found is that the exact same thing happens in mantellas, where the moms are toxic, the eggs that she carries and provisions the tadpoles with are toxic, and the tadpoles acquire toxins too through this behavior. And so to summarize that, what we found is that egg provisioning provides both nutrition and toxins. It allows moms to not only give them a nutritious protein packet, but to also chemically defend their tadpoles much earlier than what other species get or have access to. Okay, so I'm gonna sum up the main takeaways from today. So one, there are shared mechanisms, neural mechanisms involved in tadpole transport. And what's cool, about this is that we found that these specific neurons promote parental behavior in frogs, but the mechanisms are also look very similar to mammals. And so even though frogs and mammals evolved parental care independently, sometimes the same neurons are used to promote that behavior, which I think is very cool. And that that feeding circuits are modified in the evolution of parental behavior as well. So galanin promotes parental behavior, and it's also linked to resource acquisition and feeding. But even though these, there are these shared brain regions and neural mechanisms, these species-specific mechanisms do exist, and we're starting to work out how they're related to differences in life history and care strategies. But only being able to study a lot of different species that have slight variations in these different behaviors have we really been able to tease apart what is generalizable principles versus what is species-specific. And without this comparative approach, we wouldn't be able to distinguish between those two things. And then I also finally showed you that these aspects of behavior are tightly linked to the animal's ecology. And so meaning that you cannot separate the two. Okay, so coming back to our 4 questions, I showed you that there's this adaptive value of toxins, and that I didn't get to talk to you about ontogeny, which is the tadpole behaviors, which are super cool. Just to remind you, they do this begging behavior. They also are fierce cannibals and will kill each other. And so they display this juvenile aggression. And so these are other topics that my lab works on, but I wasn't able to, didn't have time to cover today. This is my lab. They're really amazing. The people that I really want to thank, though, is our local collaborators, both in Ecuador and Madagascar. We work with local scientists in the places where we do field work, and they're also our co-authors and co-PIs on our grants, and I am incredibly grateful to them for their collaboration. And so thank you. Should I stop sharing? I guess I'll stop sharing and take questions.
Thank you so much, Lauren. That was terrific.
Thank you.
Do folks have questions?
Hi, Lauren. I'm Brian Barnes. Thanks so much. Vast areas there, and I love the Tenbergen references. I'm reminded the co-location of, or close location of neurons that are substrates for feeding and then transporting the galanin neurons remind me of David Anderson's work, where triggers for aggression and sexual behavior are also co-located or anatomically close together. And I've always wondered if in humans now, if that's true, and there's evidence that it is, imbibing alcohol can cause cross-firing between those clusters, and thereby a normal behaving person who might normally separate aggression, violence, and sexual behaviors might show those behaviors together. Is there evidence that alcohol causes cross-firing between neurons?
That's a great question. I don't know. So one, I don't know about the alcohol question. There is some evidence that drugs of abuse definitely do that, other than alcohol. So for example, nicotine does that, and as well as cocaine. And so there is definitely this this interaction makes sense, because you need to adjust your resource allocation to be able to show parental care. You should stop foraging when you perhaps have a mating opportunity. And then for like mating and aggression, you don't want to attack your mate or something like that. So you need to reciprocally inhibit a lot of these different patterns of behavior to be able to show the appropriate one. And those can be tuned, yeah, by drugs of abuse. So there's a lot of work on that on nicotine, for example, and hunger circuits and social behavior, and probably alcohol too. But I just, yeah, I can't think of one off the top of my head. But definitely drugs of abuse can tune the circuits.
Mm-hmm.
Someone named Amy gonna ask a question.
Yeah, let me unmute Amy really quick. Amy, can you unmute your mic?
Yes. Thank you very much for this wonderful presentation. I just had a question on how long did it take for you to develop the relationships for those in those places to be able to study the poisonous frogs, and what would you suggest for a young researcher that would be interested in place-based research? Thank you.
Yeah, so it took me a long time to develop those relationships, because there is a long history, as in many fields, of exploitation of Western scientists coming in. So in poison frogs, for example, scientists from the National Institutes of Health poached a bunch of frogs and patented some of the chemicals on their skin, and Ecuadorians still have not forgiven them for that. And so, like, when Rafael Correa was president, he came to Harvard, and he actually like mentioned this to me when he toured our facility. And so it's definitely something still on the minds of Ecuadorian people. And so it took a long time to build trust. And so some of the things that we do is we run a lot of workshops in Ecuador. Also, almost all my grants that have Ecuadorian field work they have a subaward to our collaborators. And then they're also co-authors. So we collaborate with them from the beginning, from planning the experiments all the way to publication and finances. And so they are, we really aspire not to be parachute scientists. And what that means is that like you just drop in and get samples and then you leave. It takes a lot of, it takes years to build trust, and it takes a lot of cross collaboration and something that's mutually beneficial to both parties. So yeah, that was the advice to young people. Did that answer the question? I think that covered it, right? Amy?
Yes.
Sort of. Did I miss another part of the question there?
So yeah, so I was thinking about how applicable are some of those to be able to communicate with the government or to communicate with organizations, or did you start off with a specific community in mind, or did you already look at what was available like in the literature where previous research has been conducted? Thanks.
Yeah, so I mean, there are different stories in our lab depending on different paths in that respect based on the different projects. For the Ecuadorian and Malagasy studies, we knew that the frogs were there. We knew that they did this behavior. We reached out to local scientists for collaborations. We wrote grants together. We did field work together, and we published together. And so it was, in that case, it was us reaching out to some of them. Some of our other work in chemical ecology, so we also study how, we also work in Hawaii with nightmare fish. And so this is important to especially Hawaiian fishermen, where in certain seasons, some of the fish they get can be quite toxic. And so we're working with them to try to figure out where the toxins come from. And in that case, that was more of a project, something driven by the local community, where they reached out to some of our team members because they had a question that was important to them. And so we collaborate with them on that aspect as well. And so it depends on the project and like kind of where it originated, but we have some that go both ways. I hope that answered your question then.
Yes, it did. Thank you.
Yeah. Any other questions?
I was curious, with the different kinds of parental care within a species, do you see much variation amongst individuals? In other words, are there some females that are better at finding those pools where their tadpoles are? And is there a signal in the brain associated with that?
I don't know about, so there's definitely, I think it's hard to say because there's definitely a learned component to parental care. New parents tend to be bad parents in our frogs. And so there's some like learning that happens about, and offspring survival definitely increases with increased clutches. And so because we do a lot of field work, it's often hard to know what their experience is. And so, but in the laboratory, we definitely do see variation, at least in tadpole transport behavior. And so like whether or not they transport their tadpoles in the first place, and what we're trying to study now is how they decide how to allocate resources to specific tadpoles. And so they make decisions also. Some animals seem to feed all offspring equally, and others seem to be choosy and play favorites. And that we think that's encoded at some point in the signal of the tadpole though. And so anyway, it's hard to know. I don't think we know enough about the behavior to know what the individual variation, to the extent that we see it, what is like based on tadpole cues versus what is a learned behavior versus what's like genetically variable. So, yeah. Any other questions? I know I'm meeting with students later, so I don't know if they're saving their questions or not.
Lauren, what's been the toughest transition for you from high school through your lofty position now at Stanford?
Oh, Yeah, so I am the oldest of 6 children, and I grew up on a goat farm. And so my parents are not academics at all. And so and I went to community college for a while, and then I transferred to a university. And to me, leaving my local community and my community college and my farm was the hardest, and moving was the hardest transition for me, because I lost my social roots and my family roots. And I had people tell me that like low income community college students didn't belong at Cornell, which is where I went to, where I transferred to. And so I think dealing with the like preconceptions that transfer students or students like not from like low income areas and things like that are not prepared for university was a hard uphill battle for me personally. And then, and then I went back to Texas for graduate school, and so I went home because I was like, well, and I need to go back home. And so I went back to Texas, and then I got this thing at Harvard. And so I definitely did not think that I belonged there. And I remember walking up into Harvard my first day there being like, oh my God, what am I doing here? And I don't belong here. And so like also getting used to interacting with those people and seeing how they interacted with my like very thick Southern accent was difficult as well. And so, but definitely like the ripping of social roots from like community college to transferring was, I think, the most difficult thing for me.
Congratulations on your successes.
Thanks.